So you're going to end up with a semi-conservative model where each circle has one strand that's the old DNA and one strand that's the newly synthesized DNA, but they're identical to one another. And then all the bacteria has to do is make sure that one circle goes to each of the two daughters, and then the two daughters have exact copy of the DNA. All right? It gets more complicated in a eukaryotic cell, in part because eukaryotic cells have way more DNA. If you were to just start from one place, and they're not circular, they're usually linear. If you were to start in one place and just head out from there, it would take too long to replicate the DNA. It would take you basically a week or more to replicate all that DNA in one cell, and you need to divide much more frequently than that often. So what you do is you start these origin of replications in hundreds of, not thousands of, probably thousands of locations on a chromosome, and you start these little bubbles, okay, and you start replicating the DNA from there. All right. So again, sorry, this is just what I said again, that just blown up. That's the version that goes on in a bacteria, where you take the two circles, you take the circle, and then you start to make this bubble and replicate it, and then you get these two circles forming that are both now double-stranded. Okay. And you can, sorry, this is an EM just showing you a single bacterial chromosome in the process of doing this. So you see that that you have something that looks like this at this moment. Okay, so these are actually all double-stranded pieces here, and they're in the process of that division. The case with eukaryotic cells is a little more complicated, so you get all of these replication forks, as we call them. Okay, and you can see that in this dividing strand of DNA, they're these little bubble-looking things, and those are places where the division is taking, the replication of the DNA is occurring, and the bubble is growing. But the complication with this is that you have to think about, we're going to talk about the enzymes that are required to carry this out. Okay, because you have to have some mechanism to separate the double-stranded DNA. So normally that's very stable. Okay, and then even though the nucleotides start to line up, they don't have a backbone to them that's covalently linked. Okay, so an enzyme is required to actually zip that together. All right, so it turns out to be a somewhat complicated process in reality. Okay, and this just shows you the edge of one of these origin of replications, one of these bubble points. Okay, and there's a couple enzymes that start the process. So one enzyme is a helicase. It unwinds the DNA and turns it from a double-stranded molecule into a single-stranded molecule in this little point, in this area. And then there are proteins that come along that stabilize that in a single-stranded conformation, because the two strands are going to want to snap back together. But these proteins will temporarily keep them apart. And then you have another problem that occurs. So remember, it's a helical structure. Okay, so it looks like this. So imagine that I have this double helix, and I grab the red and green strands here, and I try to pull them apart from each other. What's going to happen to the rest of the helix? Yeah, go ahead. It's going to what? Yeah. If I do this, it's going to get more tightly wound. If I start to pull these apart, then all the coils are going to start moving. It's like when you try to uncoil a cord, right? That's a spiral. And if you try to pull the coils along, then it just gets a tighter and tighter spiral. So that's what's going to happen. So there's another enzyme called topoisomerase, which is really a pretty miraculous enzyme. It can let the DNA start to tighten, and then you'll cut it, let it uncoil, and then bind it, and then seal that break that it made. Okay, so it's relaxing the coils as it goes without letting the DNA fall apart. Okay, so it's going to go along in front, and it's going to release the tension that's being made by having to have this separation of the strands occur that's moving along. All right, and then it turns out that the enzyme you need to start the process of polymerization, it can't start de novo. You can't just have a template of DNA and a lot of nucleotides lined up and get something to come along and zip that together. It needs a little bit of double-stranded material to start off with. And so the cell has another enzyme called the primase, and there's actually little bits of RNA that line up the parts of the DNA, and they're just random bits, so they can line up pretty much anywhere. They're random little bits of sequence, so they can line up pretty much anywhere. And they sit down, and then you're going to extend off of that. Okay, so nucleotides are going to start lining up, and you're going to extend. But the other complication is that you can only polymerize a reaction of a nucleotide in the 5' to 3' direction. Okay, it's the only direction you can go. All right. Okay. So you start then polymerizing this, and again, what's going to happen is that the bases are going to line up. All right. So if you have let's say this is either the growing strand or your RNA primer, and so you have an A as the next nucleotide, right? And so you're synthesizing a new strand in the 5' to 3' direction. Okay. So the template strand is going this way, 5' to 3'. So you've got an AT paired CG, a GC, and then the next nucleotide here in the template is an A. So what's going to happen is a T is going to come along and line up to that. That's the only nucleotide that can line up in that position, and it's the one you want. Okay. And then an enzyme called DNA polymerase is going to come and cut those phosphates off and hook it to this hydroxyl group and make the bond. Okay. So now you are growing this chain of DNA as you go on that one strand. Okay. And so you just keep doing this over and over again. So what would the next nucleotide be here? A G, right? Okay, so then the G is going to come along. It's going to be the exact same thing. The polymerase is then going to connect the phosphate group that's on that to the hydroxyl, and it can keep extending that chain. But it can only do it in the 5' to 3' direction. Okay, so that's fine if we label this as the new strand going 5' to 3'. It's fine if you're going this direction or this direction. Okay, so you're growing the new strand 5' to 3'. We're looking at this little part. So that's going to be fine. So these are called leading strands in this direction and this direction, and extending them is no problem. As soon as you get a primer and you start going, you can just go on and on and on until the end of the DNA, or you reach another replication fork that's also been dividing. The hard part comes on the other side. Okay. Again, we'll talk about that in a minute. So again, just an exploded view here. So these leading strands are easy, and we'll talk about these lagging strands in a minute. Those are more complicated. So you're going along 5' to 3' on the leading strand. Okay. The problem is that you want to replicate this strand of DNA at the same time. Okay. But the new strand is headed in the wrong direction for the polymerase to work on. Okay. So the nucleotides could line up, but the polymerase can't make DNA in this direction. Okay. So what would have been nice if you could make it in both directions as the bubble expands. You could just make both strands and keep expanding the bubble in both directions. But you can't do it because of that directionality. And so we'll go through this, how you actually take care of it. And you take care of it by making short little pieces of DNA instead. All right? And so this is called the lagging strand. Okay? So these are the lagging strands. And the way they work is, again, you would want, in a perfect world, you might imagine you could also replicate the DNA in this direction, but that new strand would be going 3' to 5', and you can't go in that direction. So what instead you do is you again have the little primer sit down, and you start synthesizing in the other direction, okay? And you keep doing that, and what you do is you have a lot of primers that sit down, and you keep making little pieces, and these little pieces are called Okazaki fragments, okay? And so again, I encourage you to look at the diagrams yourself again and think about it very carefully to see why you have to do this, why it's a problem. Okay? And it's a topology problem that as you're opening up the DNA to make the copy in this direction, you've got to do it in little pieces going in the opposite direction you really want to go. And so what you do is you keep doing that. You keep making these little fragments and extending them. Okay? And then the key is then you have to connect all your little pieces together. Okay? And there are enzymes, and I'll go through those in a minute, that can do that. So as this replication bubble is expanding, the leading strand is just going straight along, but the reverse side is having to synthesize little bits of DNA to fill in as it goes. Okay? But it'll make the exact replica. It just has to do it in small bits at a time. Okay? And so what it does is something like this. So it's making the little primers sit down, and it starts to synthesize the new strand in the 5' to 3' direction. And it's using a DNA polymerase. It's called DNA polymerase 3. It's the same one that's being used on this side. But what will happen is that you'll run into the next primer, okay? And then another enzyme called DNA polymerase 1 can come along, and it can chew that RNA back out and replace it with DNA. Okay? So then the RNA primer will disappear. You'll put the right DNA nucleotides in there. And then at the very last step, the DNA polymerase 1 can't connect together the two pieces when they hit each other there. And so a third enzyme called DNA ligase finishes the job and fixes all the little gaps that are left. Okay? They're not gaps in nucleotides. They're just gaps in the bond, okay, between the last phosphate and the next hydroxyl. And so those 3 enzymes are required to synthesize the lagging strand. Okay, so by that mechanism, then you can get both of these strands made properly, the leading strand and the lagging strand. And I think this is just the same slide again, sorry. Okay, and you need these 3 enzymes. DNA polymerase 3 extends off of that. DNA polymerase 1 gets rid of the primer and replaces it with nucleotide, and DNA ligase takes care of the last step of connecting that bond that's there. All right. In reality, these things are actually ordered into a sort of a complex of proteins. Don't worry about this. This is just to show you that the reality is a little bit more complicated, that there's actually other proteins that connect things like the helicase and the polymerase together. So everybody moves along together on the leading strand as they go. Okay? And that keeps things sort of coordinated, and it lets the DNA, after it's replicated, wind back up. All right? So that's how you're going to accomplish it. So how fast can polymerase go? So polymerase is actually very, very rapid. It can go, it can synthesize this reaction at a very high rate. The problem also, though, is that it needs to be very faithful. So it can't make mistakes. Okay? Because if it makes mistakes, then you're going to get the wrong nucleotide in that position. And we'll talk more, a little bit about the genetic code. That in genes, you take that DNA template, use it to make RNA, and eventually you read that code into protein. And the nature of a lot of mutations is that you incorporate the wrong nucleotide. Okay? And then you have a mutant protein because it leads to the wrong triplet codon in the RNA, the wrong protein at the end. So how faithful is it? So in your cells, it basically makes a mistake only once every 10 million base pairs. Okay? 10 to 100 million base pairs. So it's very high fidelity. Okay? And the reason it's very high fidelity is it actually checks for mistakes as it goes. So the DNA polymerase 3 is actually 3 proteins together in a complex. And the function of one of those proteins at the back edge as it goes along is that it basically, in a sense, feels the shape of the DNA. And if the wrong nucleotide's been incorporated, it can see that the double helix, that the helix being made is not properly shaped. And if that happens, the polymerase will back up, it'll cut out the mistake, and then let the right nucleotide come in, and it'll start again. Okay? And so it has very high fidelity. And so if you eliminate that, the component that actually is responsible for the fidelity, what happens is the polymerase becomes even faster, but now it has a much higher error rate. Okay? And so there's actually a balance between how fast you want to go and how faithful you want to be and not make any errors. Okay? And then even if it leaves an error behind, there is actually a system to fix anything. So there are other enzymes that control the DNA, and they look for these little bulges that indicate that the wrong nucleotide has been put into a strand. Okay? And they'll come along and fix that. Okay? But can you see a problem with fixing it after you've made it? What's that? Okay, well, that's a biochemical problem, but yeah. What's that? Okay, so what you're trying to do is, that's right, you're trying to keep the mistake from being passed on. Okay? So you're trying to detect the mistake that you made and now cut it out and replace it with the right thing. Yeah. Yeah, exactly. So the problem is that if you do it while you're replicating the DNA, because of the way it works, the polymerase knows which is the template strand and which is the new strand it's making. So if it finds a mistake, it knows that the mistake is in the new strand, right? And that's what it needs to correct. If you find it after the fact, it's a little complicated for the cell to tell which is the new strand of DNA and which is the old strand of DNA, because now they're both already all polymerized together. Okay? It isn't going along creating this. And so it can cut out a mistake and replace it, but the problem is that it doesn't necessarily know which way to replace it. Okay? And so at that point, then both strands can have the same mutation, so they can either get fixed or have a mutation. Yeah. Yeah. Okay, so right, we have to be careful not to anthropomorphize so much. So because of the topology of the way the polymerase is moving along, it's holding on to the template strand in a particular way. And so the part of it that can fix the mistake is only contacting the new strand that it's making. So if it fixes the mistake, it can only, if there's a mistake that's there, the only excision it can make is in the new strand. Okay? But if you do it after the fact, you can't tell which is the new strand and the old strand. Okay? There are some tricks. It turns out that you can make modifications to the nucleotide as you go every now and then on the new strand versus the old strand, and an enzyme can come along and still actually figure out which is the old and new strand. Okay? But it's pretty limited. And so a lot of times the mistake will get made. In the end, the whole system is incredibly faithful. So in a replication of all of those, 3,600 billion nucleotides in your cell, in a round of replication, you'll only have, like 100 errors. Okay? So it's remarkably faithful. But those 100 errors could be a problem still. Okay? But if you had no errors, it would probably take you so long to double check over and over again. It would be too slow a process. So anyway, so that's how the system works. Okay? There's one last problem that you run into. We're almost done here. And that's when you get to the very end of the chromosome. Okay? So you're going along, and you have no problem with the leading strand. The leading strand can keep synthesizing DNA until it gets to the end of the template. But you end up with a little bit of a problem in the lagging strand. So you can't actually fill this piece in. Okay? Because you don't have any DNA on this side to act as a template for the nucleotides to line up. And the nucleotides can line up, but there's no way the polymerase can get in there. Okay? So you run into a problem. It means that every time you replicate your chromosome, if you have a straight linear chromosome, okay, and again, bacteria have circles, it's not a problem. But for you have linear chromosome. It means that on every round, you lose the very end of the DNA, okay? A little bit at a time. But that adds up over time. So then when you make another replication, your template here, right, doesn't have any of this information, so it gets made like this instead. Okay? And if each round you go through, you lose a little bit off of the end. So in fact, what happens is that your chromosomes have a long series of repeats that don't actually have any functional significance. They go off the ends of the chromosome. And so in every round of replication, a few of them will get lost. Okay? But there's no important information there, really. So you can do that. But one idea is the reason that your cells age is because eventually they run into a problem. They run out of that little repeat that they can afford to forget about on each replication. And then they start to chew into the essential part of the chromosome. And so that's one idea about what contributes to aging. It's clearly not the whole explanation at all. But it's a popular notion. And so you see these, if you look at human chromosomes, you see on the tips are these repeat structures. They're lit up here by a technique to detect that particular sequence. And they're on the ends. And it does turn out that there are enzymes called polymerases that help to protect the ends as well and to put new repeats on to the ends to try to protect it. And that's very important for your germline. You don't want to pass on chromosomes to your offspring that are already shortened because they're going to have a problem right away in the next generation. And if we talk about cloning later in the course, remember this, because this ends up being a problem. If you clone an animal from a somatic cell, its telomeres, those ends of the chromosome are a little shorter. And so if you keep doing it, you're going to have a problem eventually. All right? So the cells have a way around that. And then just to end at the very end here, the DNA in reality has a more complicated structure. So we talked about it being a double helix, okay? But if DNA were just a double helix and it were billions of base pairs long, it would be meters and meters long, okay, inside a single bacteria. But it's not, and that's because it coils even tighter and tighter. And so this just shows you some of what happens. There are proteins called histones, and the double helix wraps around those in yet another helical structure. And then those histones get together and they actually wrap around themselves into an even more complicated structure. And so the next thing you know, you take a strand of DNA that would otherwise be, many meters or even more long, and you coil it up into really, really tiny short chromosomes, okay? So, and that's important to the structure, and then that's what you actually see when you look in, say, human cells. You see these chromosomes. So we'll go ahead and end there, and I'll pick up with PCR in the next lecture.